I'd like to thank everyone who's attended the webinar. Hopefully through the slides you'll see, you'll be convinced that this technology can be applied both in analytical development and hopefully eventually in QC as well, to ease the process of technology transfer from one department to the next, and also to simplify flow cytometry workflows in general. So I'll start by giving you a little bit of background about who Assay is. So we are developing an off-the-shelf gamma delta T cell product, which is composed of engineered gamma delta T cells with CARs, TCRLs, or TCRs. We are developing a pipeline of products that range from liquid and solid tumors. We've currently developed a robust large-scale manufacturing platform. Key here is that we have a strategic collaboration with Regeneron Pharmaceuticals. Where Assay comes in is we actually have a process that starts with a normal healthy donor. And from this normal healthy donor, we can activate specific subsets of gamma delta T cells, engineer them to express CARs or TCRs, expand them to the point where we can actually create multiple doses from one incoming leukapheresis. Now, of course, as this is a cell therapy product, flow cytometry plays a role in a lot of different aspects of the manufacturing process. In this slide, I highlight 3 points where flow cytometry is required to understand what we're manufacturing. So at the beginning, we use flow to identify the subsets of gamma delta cells that will be expanding. Post-expansion, we want to make sure that our cells have been properly engineered and that the purity of the material has reached the desired level. And then after cryopreservation, the material is thawed, and again, the same analysis is done to verify the batches for release for actual treatment of patients. So at least the goals in my particular group at Assay are to create reproducible assays that will guide process development and eventually the manufacturing process as well. Now, the key here is that we need to turn the art that is flow cytometry into something that's reliable and reproducible. So we quickly decided on several different aspects of trying to lock down the flow cytometry assays that we were creating. So optimize the panel design, titrate antibody concentrations, lock the cytometer settings to minimize missing drift, create and use cell product reference material, and I'll show you some of that later on. Of course, develop SOPs for staining to minimize operator variability, and use analysis templates or automate the analysis completely. So the one outstanding point here that was lacking was the fact that we were still doing manual centrifugation. So these were the first steps that we knew of until we realized that the DA cell existed. So to give you a sense of the complexity of the flow cytometry panel that we're currently using, we use an 8-color panel run on a Canto 2. We're actually using 10 different antibodies in the panel to be able to identify different aspects of the material, with particular focus on the gamma delta T cells, which we can identify by a TCR gamma delta antibody, also through CD3. And then we have 2 gamma delta subsets that we're interested in, V delta 1 and V delta 2. And then we can look at impurities in the material, including B cells through CD19, monocytes through CD14, alpha beta T cells through TCR alpha beta staining, and NK cells with CD56. Now, the thing you'll notice here is that the gamma delta frequency in starting material, which is what's shown here, is pretty low, less than 2% of starting total T cell fraction. So we need to be able to have high resolution on this particular population. So prior to implementation of the DA cell, we did take a look at the donor variability of the material, and you can see that 3 different donors, there's high variability in a lot of the starting populations. Most of the starting material is high in monocytes and TCR alpha betas. You can see here that the gamma delta population within all live cells is relatively small. Okay, we're somewhere in the order of, again, less than 2% of all incoming material, and we need to have clean resolution on this particular population in order to make sure that we're manufacturing the right cells for treatment of patients. How do we compare a manual centrifugation method to this new DA cell? So manual centrifuge procedures are still kind of the standard spin and flick. So add buffer, spin it down, flick off the buffer, resuspend the pellet with addition of buffer, and go through at least 2 rounds or so of the wash procedure. The DA cell, by comparison, is load the plate, push a button, walk away, come back when it's done. Not that you have that much time, because it's only about 2 minutes per wash cycle in terms of a comparability for the manual centrifugation method. So the first thing we decided to do with Curax's help is to map out our workflow and see what our time savings would be if we implemented the device. So you can see our staining procedure in the middle, with emphasis particularly on the wash steps in the gray. And then on the left side is the timing for the manual centrifugation method. And you can see per wash cycle, it's roughly about 10 minutes, and we do 2 washes between each of the staining steps, which means our total stain time is, wash time is about 60 minutes. Whereas on the DA cell, every single cycle for washing is fixed at 2 minutes, which means we only spend about 6 minutes of time per entire flow cytometry workflow waiting for the washing procedure. Now, in terms of the data quality that we received, so this is representative data. On the left-hand side, you can see manual centrifugation, where we're comparing this V delta 1 versus the V delta 2 subset. And on the right side, you can see the DA cell. And there's already some striking visual differences between the two. So if you look at the V delta 1 single positive population in the upper left, the DA cell material consistently comes with less spread and less fanning of that single positive population. So we get a cleaner type population. And the V delta 2 single positive in the lower right tends to be tighter. Now, the added benefit here is it makes the gating strategy a lot easier for operators because the separation between the negative population and the positive population is that much cleaner. We did a head-to-head comparison between the two methods with 2 different donors, and here I'm highlighting 4 different populations of cells. And yes, there is a little bit of variability in the frequencies, but it's nothing that we would consider outside the normal noise level. So the reported frequencies that we had, at least for the 4 populations that I'm depicting here, are within the noise that we would expect for normal variability in flow staining. We decided to dive a little bit deeper. So what we did here is we gated individual populations of cells. And because the panel uniquely identifies the populations through 1 marker, we looked at the mean fluorescence intensity of each of the individual markers in that population. And then we plotted the MFI between the manual centrifugation on the left and the DA cell on the right. And what's striking here is that the DA cell and the manual centrifugation method gives us roughly equivalent MFIs with a slight advantage to the DA cell. The key for me here is that there was no deterioration in the signal with implementation of the DA cell. So that data set was enough to convince us to bring the DA cell in and to use it in our workflows. So the next couple of slides, I will show you the performance of the machine across several runs using an internal reference control that we've developed in-house. So what I'm showing you here are 2 independent operators doing 2 independent stainings with replicates for each operator. And you can see that the DA cell allowed us to get very tight control over the populations that are reported. You can see that we have very tight CVs, again, largely in part to the fact that there's clear separation between the populations. And with centralized analysis, you can see that we can get the CVs down to 10 to 11%. And that 10 to 11% CV range is on populations that are less than 1% of the total live cells in the starting material. So what I'm showing here is kind of the reproducibility of this reference material over time. So again, we use a frozen bank of cells that we can thaw for every single run. And you can see over 12 runs that now span almost 4 months of operation, the relative frequencies of each one of these subsets, while there's some fluctuation, overall the trends are fairly consistent across the board. And this is highlighted again, the optimization that we got with implementation of the DA cell. So where are we now? So implementation of the DA cell currently saves us about 50 minutes per assay run. So even over the course of 1 week, where we're running, maybe 10 assays between all the individual users, we're talking about almost 8 to 10 hours of FTE time that's saved just in terms of optimizing the wash step. So the data generated from the DA cell is at least equal to the data generated from the manual centrifugation method. There's a slight advantage to the DA cell in terms of the MFI. The frequencies are roughly equivalent. Key here is that implementation of the DA cell did not require major changes to our existing procedures. It was very plug and play. We could swap out the centrifuge, bring in the DA cell, and we did not have to modify our staining workflow in any significant way. And I think I've highlighted the fact that the use of the DA cell does minimize operative variability. So currently the DA cell is part of our workflow. It's being operated in analytical development, and all of our subsequent flow cytometry based assays will be developed using the DA cell as the wash method. We are currently in the process of tech transferring this particular workflow into QC, and we think by having the DA cell in place that will simplify the process because we're transferring a workflow from a device to a device rather than from an operator to an operator. And with that, I'd like to thank the people at Assay that actually did the work, so Praveen and Judith, and Carriox for their support as we transition from our manual process into the automated process. Thank you.